Hello and welcome to today's Journal Club, in which we will discuss two recently published papers that explore the role of mosaic mutations in autism. I'm Claire Cameron, Engagement Editor at Spectrum, the home for autism research news and analysis. We'll begin today's presentations and discussion in a moment, but first, know that we'll be fielding questions at the end of today's presentations, but you can ask them at any point during the session. To ask a question, submit them via the chat window on your screen. We encourage discussion among the audience throughout the presentations. And as always, I'll note that if there are any members of the press tuning in, you can only report information presented during this Journal Club if that material has already been published elsewhere, or if you have first obtained express written permission from the presenter. Today's first presenter is Rebecca Barnard, who joins us from Portland, Oregon, where she is a postdoctoral researcher in the O'Rourke Lab. Barnard received her BS in Biology and Biotechnology from James Madison University in Harrisburg, Virginia. Afterwards, she moved to Fort Collins, Colorado, where she spent 2 years at the Centers for Disease Control as a serologist for plague and tularemia diagnostics. She then attended Colorado State University for a PhD in cell and molecular biology under the mentorship of Dr. Daniel Gustafson, studying the autophagy pathway in the context of cancer treatment and development. Today's second presenter is Elaine Lim, who joins us from Cambridge, Massachusetts, where she is a postdoctoral fellow at the Weiss Institute for Biologically Inspired Engineering and Harvard Medical School. She received her BS in Computer Science and MS in Bioinformatics from the National University of Singapore, and PhD in Genetics and Genomics from Harvard University. She seeks to understand the genetic causes of autism through the discovery of novel risk loci coupled with in vitro screens to elucidate causal mechanisms. Welcome, Rebecca and Elaine.
Thank you, Claire, for the introduction and also for the invitation to present our group's recently published work entitled Exonic Mosaic Mutations Contribute Risk for Autism Spectrum Disorder. So first, I just wanted to give a brief background on autism genetics and mosaic mutations just to provide some context for our studies. Autism risk has a strong genetic contribution, but identifying risk factors has proved to be very challenging. We've discovered that there is a great deal of heterogeneity, with risk factors ranging from single nucleotide changes to large structural variants. And there have been hundreds of implicated loci. One approach to reducing some of this complexity is to look at simplex families, where there is no family history and the disorder is newly appearing. In this scenario, we are enriching for de novo variants and likely more severe contributory variants as well. And we expect about 1 mutation per generation, and mutations are unlikely to occur in the same gene by chance. So if multiple occurrences in unrelated individuals are observed, this boosts our confidence in the gene as a risk factor. And the Simon simplex collection is the largest cohort of ASD simplex families, about 2,500 families in total. It is one of the most well-characterized ASD cohorts in terms of extensive phenotypic and clinical data. Exome sequencing studies, that is sequencing just the coding portion of the genome of the SSC cohort, has been transformative in our understanding of ASD genetics. And from these studies, 27 to 35 genes have been identified as high confidence risk genes. We also find that de novo copy number variations, likely gene disrupting mutations, which include nonsense, splicing, and indels, as well as missense mutations, make up 30 to 50% of the contribution to ASD. But what about the rest? What else are we missing? So the current focus of risk factors has centered on germline de novo mutations, which are present at the time of fertilization. However, recent studies have implicated mosaic mutations, those that occur after fertilization, in neurologic disorders. There are also examples of brain-only mutations where the variant is not detectable in blood, possibly representing a later embryonic event. Mosaicism may also be one explanation for why we see variability in presentation, including subtle characteristics witnessed in parents of patients with autism. Transmission of mosaic variants is also an underappreciated phenomenon that can have important implications for recurrence. Therefore, mosaicism may be another genetic contributor to autism development. And so mosaicism is when you have a mixed population of cells, some having the mutation and others not. When we look at genomic data, a classical germline mutation or variant would be present in all cells and would make up 50% of the fraction in DNA. And it can either be inherited or it can happen as a spontaneous event. As a mosaic event would occur after fertilization, the fraction would not be 50%. The fraction depends on when and where in development it occurs. And as the fraction can be very low, mosaics can be hard to detect, and distinguishing true mosaics from germline is very difficult. Single-cell sequencing of neurons in some studies suggests that mosaicism is widespread, including populations with copy number variations. Therefore, we are likely either missing or incorrectly attributing mosaic variants as germline, leaving the role of mosaicism as still poorly understood in the development of autism. So first, we wanted to see if we could find any de novo mutations that have evidence of being mosaic. And we reevaluated about 5,000 some previously published single nucleotide variants, or SNVs, using existing SSC data. And these SNVs were originally identified using methods tuned to find germline events, not necessarily mosaic. And we used a binomial approach to identify likely mosaics. So those with an alternative allele count significantly deviating from a heterozygous 50% allele fraction expectation. And we found that about 11% of the de novo mutations show evidence of being mosaic. We also find significantly more putative mosaic variants in the de novo versus rare inherited SNV population. Sampling the rare inherited SNV population over 10,000 trials, we on average find only that 3% of rare inherited SNVs show a mosaic-like allele fraction, suggesting we are finding real mosaics in the de novo SNV population. So if we are not recognizing these as mosaic mutations, what others might we be missing? We know germline methods are not sensitive to low allele fraction, so these could represent just the tip of the iceberg. We need better methods, ones that are tuned to mosaic variants that can identify variants in this allele fraction region. So for our general study approach, we needed to develop a computational and empirical method to detect mosaic variants and distinguish true mosaic events from germline. In a general overview, we first evaluated a number of callers specifically tuned for mosaic variants, not requiring matched tissue, using simulated data. And based on their performance at different depths and allele fractions, we decided on VARSCAN2 and LowFreq2, along with our own custom script we called MPUP for MPilot Parser. We then used these callers on a subset of the cohort we called our pilot families. And we validated these predictions in a high throughput and highly multiplexed method using small modified molecular inversion probes. And we used this data to inform us about ways to better distinguish germline variants from mosaics. And we also developed a logistic regression model to better predict true mosaics. We then did 2 more rounds of validation using a larger independent subset of the cohort to train our model, finally arriving at an approach to score variants within the cohort for likelihood of being a true mosaic. And as I mentioned, we validated several different rounds of data, first on validated calls from 24 families, then on an independent set of 400 families, 78 of which we validated. And from this process, we also developed some additional heuristics that helped us to further distinguish germline and mosaic, including setting a hard threshold for any variant that had an allele fraction with a confidence interval overlapping 0.4, removing reads with mismatches of 3 or more, removing sites and segmental duplications or tandem repeats, a cohort count greater than 2, and an alternative allele fraction greater than or equal to 5. So with these new filters and model, we did a third independent set of validations. We were ultimately able to achieve a sensitivity of 0.94 and a positive predictive value of 0.85 with a model score of 0.518. And we also arrived at a joint coverage threshold of 45x and an allele fraction of 5% or greater for our high confidence set of variants. And using this approach, our goal is to perform a systematic evaluation of the mosaic mutations and determine the rates between parents, children, and transmission. So after applying our model to the cohort, we identified a total of 2,147 de novo mutations in children, and of these, about 22% are mosaic. We identified 1,293 total mosaic mutations in parents, which is a 2.5-fold greater burden than children. However, we believe some of this excess may be due to incomplete filtering. Unlike the children, we do not have the prior generation, that is the grandparents, to compare to in the parents. So extrapolating from the application of transmission filters in the children, we estimate that 40% of parental calls are in excess, bringing the rate down to only 1.5 times greater than children. We found that 7% of parental mosaics are transmitted, and this includes sites previously reported as germline de novo in the children. We also identified obligate gonadal mosaics in the parents. We could see the same apparently germline de novo variant in both offspring, but no evidence in either parent. And we estimate that 6.8% of presumed de novo mutations in children have evidence of parental transmission. And so comparing to the previous data, we find 332 new, not previously published mosaics in our high confidence call set. And of the published de novo variants we initially flagged using the simple binomial approach, only 138 remained mosaic. So when looking at the allele fraction distribution of our mosaic variants, we observed that the parental transmitted distribution skews high. And the higher allele fraction is consistent with an early embryonic event, which would be necessary if the variant is to be present in both blood and gametes. And when we look at the same variant in children, we find an allele fraction consistent with a germline distribution, suggesting these sites are not systemically biased, so they don't appear mosaic in every generation. Therefore, we think these are real mosaic events, and the higher fraction suggests they occurred as an early embryonic event. We see a large number of mosaic events in this allele fraction range in the children, indicating that they are likely early embryonic events as well, and these make up about 30% of all mosaic calls in the children. We also looked at the mutational spectrum using the frequency of each type of mutation and its context within each possible 96 trinucleotides. And as seen in many other studies, we observed the highest frequency at CpG dinucleotides, and the spectrum for parental mosaics looks nearly identical. And the spectrum also looks the same whether we split out probands or siblings, mother versus father. It is also very similar between germline and mosaic variants. So the same mutational processes are driving germline and mosaics, as well as proband and unaffected siblings, parents, and children. And a recent study using a large cohort of different cancer samples identified 30 unique mutational signals representing different mechanisms driving mutation. And we find that our spectrum for germline, child mosaics, and parental mosaics all most highly correlates with cancer signature 1. And the signature is associated with endogenous mutational processes. We also saw a close association with cancer signature 6, which is associated with defective DNA mismatch repair. And next, we wanted to determine if probands had an excess burden of mosaic mutations. And we were also interested in looking at burden across different allele fraction windows. Burden may differ based on embryonic timing, particularly those high allele fraction early embryonic events could contribute more substantially. So we set a threshold for the minimum coverage using a probability model to determine the reads needed to detect the variant and removed families not meeting the joint coverage threshold required for each allele fraction. And unexpectedly, we saw that probands had a significant excess of synonymous variants, those that are supposed to be silent and not affect protein function. And this is across multiple allele fraction windows. Looking at the 12.5% 50x window, we determined that probands have 0.046 events on average when extrapolating to the full exome. Their unaffected siblings, meanwhile, only have 0.023 events. And the differential is 0.023, suggesting that 50% of synonymous mosaics contribute to ASD. And this translates to 2.3% of simplex cases within the cohort. And so typically we consider synonymous variants to be relatively benign, but some recent studies show synonymous variants may contribute to human disorders, including autism. And in cancer, it was demonstrated that synonymous variants in oncogenes can impact splicing and may act as cancer drivers. One possible way synonymous variants could impact protein function is affecting alternative splicing. A mutation arising in a splicing regulatory motif can prevent the binding of a splicing enhancer or repressor, altering whether or not that exon is included. Additionally, these regulatory regions are enriched near the ends of exons where the canonical splice sites lie, about 30 to 50 base pairs from the exon flanks. The mutations closer to splice sites may have a stronger effect on splicing. And when we looked at the distribution of the minimum splice site distance in our synonymous mosaic variants, we do in fact see that the synonymous mosaics in probands are significantly closer to splice sites than either their parents or siblings. We also used multiple in silico approaches to predict the variant's effect on splicing, namely whether the variants impact the splicing regulatory region. And we used Human Splice Finder, which uses a collection of different splicing algorithms. And given the high call rate of splice-affecting variants, we removed the two most frequently called matrices and reclassified. We also used an independent algorithm called Spanner, which takes a machine learning-based approach. We found that probands are significantly enriched for synonymous mosaic variants impacting splicing as compared to their unaffected siblings, including when restricting to variants falling within 50 base pairs of the canonical splice site. We identified synonymous splice-affecting variants in embryonic brain-expressed genes in probands who do not have any other non-synonymous germline mutation. So if validated for differential splicing, these may represent novel candidate risk factors. Now looking at missense variants, ones that actually affect the protein, we do not see any burden looking at the whole cohort. And this is not what we see in germline missense. So since a large fraction of the SSC cohort actually has a contributory likely gene disrupting or LGD mutation, we thought that looking at families that do not already have a germline LGD or any non-synonymous mutation would improve our chances. We also reason that probands may differ in the types of genes that are affected. Other studies have demonstrated that probands also have increased burden in genes that are constrained or intolerant to mutations. Therefore, we think the signal may be stronger in genes intolerant to mutation, as has been shown in the germline mutations. And we used a list of essential genes which are associated with lethality in mice and genes with low frequency of loss of function mutations in the exact database. And when we divided the cohort and looked within essential or intolerant genes, we do in fact see an increased burden. And this is particular to probands who do not already have an LGD or any non-synonymous mutation. And we estimate that 52% of missense mosaics in essential and about 60% in intolerant are contributing to autism. And this accounts for 1 to 2% of probands who do not have germline LGDs or non-synonymous mutations, and this would be about 1% of the whole cohort. So for germline LGD mutations, enrichment was seen in different gene sets that are relevant to autism, including chromatin modifiers, embryonically expressed genes, essential genes, postsynaptic density, and FMRP-related genes. Similarly, missense germline mutations were enriched as well. And in our germline calls, we see recapitulation of FMRP enrichment. However, for our missense mosaics, the only enrichment we observed is chromatin modifiers. And while nominal, this enrichment is still a twofold difference compared to siblings. And so lastly, we looked at the overlap of genes targeted by mosaics and germline mutation. And we used a list of the most high confidence ASD risk genes, as well as for ones identified in other developmental disorders. And for this analysis, we also included previously published indels and SNVs that did not quite make our 45X cutoff, but passed all other filters. And we identified 12 mutations occurring in these high confidence risk genes as compared to only foreign siblings. And these included 3 novel events not published and a novel mutation in SCN2A that was transmitted to the proband from the mother. And so we also observed mosaic mutation in genes that overlap additional proband non-synonymous germline mutations. We found 7 mosaics that overlap with germline LGDs. These include novel nonsense mutations in BAZ2B, UNC79, and USP15. We also find 10 mosaic mutations that overlap with germline missense. And many of these mutations are in chromatin remodelers, including BAZ2B, SSRP1, as well as transcription factors and synapse-related genes like UNC79. And based on these additional mosaic calls and their roles in neurodevelopment, we think these genes may be novel candidate risk factors for autism. And so in conclusion, we developed a highly robust multiplexed approach for identifying and validating mosaic mutations. We estimate that mosaic mutations contribute to 3 to 4% of simplex ASD, with 2.3% coming from synonymous mutations and 1% from missense mutations in intolerant or essential genes, particularly for probands who do not already have germline LGD or nonsense mutations. We see evidence of parental transmission and think that these account for 6.8% of presumed de novo mutations in children. We also see overlap of mosaic mutations with germline mutations in high confidence and candidate ASD risk genes. And so with that, I would just like to thank my other co-authors and for everyone in the study of the original SSC data sets, and we'll turn over the presentation to our next speaker, Elaine. Thanks.
Yeah, thanks, Rebecca. So Rebecca has given a really nice introduction to the topic, and so I'll delve directly into our data. So we took essentially a very similar approach, but other than using the data from the SSC, we had access to data generated by the Autism Sequencing Consortium as well. So in all, we explored mosaic mutations in about close to 6,000 trios from both data sets. And for a start, we categorized all the mutations that we detected into 3 different groups, which we termed Group A for all de novo mutations. Group B are a subset of the mutations, of the de novo mutations that we think are putative, and these had skewed allele fractions of less than 80% of the modal allele fraction. And Group C, we define them as a stringent category or high confidence group of mosaic mutations using more stringent criteria. So the diagram on the left shows the schematic. In all, we detected about close to more than 4,800 de novo mutations, out of which we estimate that 23% of them are putative mosaic mutations, and about 9.7% of these are likely to be high confidence mutations. When we compared the mutations with previously published data, given that many of these exome sequencing data were published back in 2014, we found that out of the mutations in Group A, most of them were previously reported. So 73% of these mutations had been previously reported in 2 prior publications. But when we looked at our high confidence group of mosaic mutations, we saw that only about 16.7% of these mutations were previously published, and in fact 83% of these mutations are novel to our study. And what this suggests is that most of the prior studies have focused primarily on the germline de novo mutations and might have missed calling some of these mosaic mutations. So to confirm that we are detecting real mosaic mutations, we performed a large-scale resequencing set of experiments across hundreds of different mutations in 3 different phases. And we used 3 independent resequencing approaches: clone-seq, pyrosequencing, as well as targeted PCR with MiSeq. And in all cases, we found that the Group C mutations do indeed show skewed allele fractions ranging from 85% to 97% validation rates across the 3 phases. In addition, we wanted to estimate as well how many of these Mosaic mutations, especially those found in group C, which is our high confidence group, are actually genuine de novo mutations. That is, they are not present in the parental DNA. And for that, we performed standard sequencing on 82 mutations and found that 89% of them are validated as genuine de novo mutations. So taken together, the 2 different validation rates, we estimate that somewhere around 7.5% of all de novo mutations detected from the exome sequencing data are likely to be true post-zygotic Mosaic mutations. So we compared the properties of these Mosaic mutations with the germline de novo mutations found in the same data, and we saw certain properties that mimic the somatic mutations that have been observed in cancer genomics. For instance, the Mosaic mutations that we detected in blood are enriched on the antisense or the non-transcribed strand, similar to somatic mutations that have been found in cancers. And we did observe certain properties, genomic properties. For instance, the Mosaic mutations are enriched for A to C and T to G mutations compared to the germline de novo mutations. However, in cancer, for somatic mutations that have been reported in cancer, there is a strong correlation of these somatic mutations in regions of the DNA with late replication timing. However, while we saw a modest excess of our Mosaic mutations detected from blood, we did not see a significantly strong excess. And germline mutations have been previously reported to be enriched on the paternal compared to the maternal chromosome in ASD. And when we looked at the Mosaic mutations, we did not see any excess of these mutations on the paternal compared to the maternal chromosome, suggesting that the Mosaic mutations are equally likely to occur on either the paternal or the maternal chromosome in the embryo. When we looked globally at the overall properties and rates of Mosaic mutations in the probands compared to their unaffected siblings, similar to what Rebecca has presented, we did not actually observe any significant excess of deleterious Mosaic mutations, either loss of function or missense mutations globally in the probands. However, when we looked specifically at the genes that were impacted by non-synonymous Mosaic mutations, we saw that in the probands, there were 735 genes that had at least one non-synonymous Mosaic mutation compared to 322 genes in the siblings. But the number of siblings in our study was a lot smaller than the number of probands as well. And when we looked specifically at genes that had recurrent, so more than one non-synonymous Mosaic mutation, we saw that there were 27 such genes out of the 735 genes in the probands, and 2 out of 322 genes in the siblings. After correcting for the difference in the numbers of probands and siblings, we still observed a 6.1-fold excess of genes with recurrent non-synonymous Mosaic mutations in the proband. And this suggests that while there isn't a global excess in the rates of Mosaic mutations or deleterious Mosaic mutations in the probands, some of these genes, especially those with recurrent non-synonymous Mosaic mutations, are likely to contribute to ASD in a subset of these individuals. To tease out the signal, we turned to an approach that was previously described by Mohammed Udin and Steve Scherrer in 2013, where they looked specifically for exons that are highly expressed in the brain using public data from the BrainSpan project, but yet these exons are depleted for deleterious variants from a large control population, such as the 1,000 Genomes project. So when we overlaid the data from the Mosaic mutations as well as the critical exon analyses developed by Muhammad et al., we found that the Mosaic mutations that are high confident in group C are strongly enriched in prenatal brain samples, in genes that are expressed in prenatal brain samples but not in postnatal brain samples. In addition, we looked at the distribution of Mosaic mutations in genes that are expressed across 16 different regions of the brain, and we saw that there is a very strong excess of Mosaic mutations in genes that are found to be expressed in the developing amygdala compared to other regions of the brain in the BrainSpan project. So we further scrutinized what are some of these genes with recurrent non-synonymous Mosaic mutations, and can they teach us something specific about ASD? So over here in this table, I list just the top 10 genes with recurrent non-synonymous Mosaic mutations. And one of the genes that we found really interesting was SMARCA4, where there are 3 probands with autism with putative Mosaic mutations in SMARCA4. And we were able to obtain fresh blood samples from these 3 individuals for validation. Using Sanger sequencing, we confirmed that the 3 mutations indeed arose de novo. So they are present only in the probands, but not in either parents. And using subcloning followed by Sanger sequencing, we confirmed that 2 of the 3 mutations are indeed arose post-zygotically, whereas the last mutation, P109L, is likely to be a germline event. So SMARCA4 is a really interesting gene, and this is the mutational landscape of the protein itself. While biallelic truncating mutations have been reported in this gene to be associated with cancer, the mutations are indicated in red above. We found that, on the other hand, many mutations, primarily missense mutations, have been reported in individuals with Coffin-Siris syndrome, indicated in blue below. And because many of these mutations that were previously reported in Coffin-Siris syndrome, which is marked by severe intellectual disability, appear to be missense mutations, and almost no loss of function mutations were observed in these individuals, it has been postulated that the mutations that are found in individuals with Coffin-Siris syndrome are probably gain of function or dominant negative mutations as opposed to loss of function mutations, like the ones observed in cancers. The 3 mutations that we found in the autism probands are missense mutations, highlighted in green, and all 3 mutations happen to lie in the N-terminus domain in a region that binds a protein called CREST. So we were able to get extensive phenotyping data from these 3 ASD probands, and we found that they had high IQs of greater than 70, so there is no evidence of intellectual disability in these 3 ASD probands, and they do not have the typical features associated with Coffin-Siris syndrome. So they appear to have idiopathic autism. So SMARCA4 itself encodes the protein BRG1, and BRG1, at least the N-terminus domain, when we looked up prior literature, we found that the N-terminus domain of BRG1 interacts with another protein called CREST, and it represses CREST-mediated transcription. And one of the downstream transcriptional targets of CREST is actually NR2B, which is encoded by the green2B gene. Now, green2B has been defined as an ASD risk gene, and so this provided us with a very testable hypothesis that we wanted to follow up. That is, the 3 mutations in the ASD proband in SMARCA4 would probably result in green2B expression. And for that, we mutagenized 2 out of the 3 different mutations and overexpressed them in mouse neuroblastoma N2A cells, and found that indeed the overexpression of the 2 Mosaic mutations resulted in significant significantly lower expression of green 2B in these mouse cells, suggesting that the mutations that we have detected in SMARCA4 are probably pathogenic and that they do contribute to autism risk, probably by mediating green 2B expression through CREST. So we'd like to acknowledge, of course, this work would not have been possible without a lot of collaborators, including Chris Walsh and the fellow lab members in the Walsh lab, Joe and Sylvia, Steve and Mohammed, and of course George and Rigel. And the data is obtained from the Autism Sequencing Consortium as well as the Simon Simplex collection. And we are open to questions, I guess.
Thank you both, Rebecca and Elaine. We do have a couple of questions, and I also want to give you both time to ask questions of each other because I'm sure that you do have questions for one another. Our first question, which is directed at, well, I suppose it's directed at both of you, is that the Simon Simplex collection only collected blood for their genomic analyses. So how can you tell that there are mosaics based only on blood? Wouldn't you have to collect other tissue types? And are you just looking at mosaics in the blood?
Should I answer that question first?
Sure. It's up to the two of you how you want to answer.
Okay. So we are looking only at the mosaics in blood, and because of that, there are certain limitations and assumptions that we make as well. For instance, we would assume that some of these mosaic mutations that do contribute to autism are first of all detectable in blood, so they have to happen at a very early stage before the blood and brain lineages diverge. However, there are other ongoing studies that make use of postmortem brain samples, and hopefully they could tell us something about brain-specific mosaic mutations.
Okay. Just following on from that, Rebecca, I know from our reporting on the study that this is something that the Aroke Lab is looking into now.
So we are not necessarily getting brain samples from autism patients, but we are looking into other disorders such as focal cortical dysplasia and seeing if we can potentially detect brain mosaics in the brain in saliva or possibly blood samples. So yes, that is kind of some ongoing research we have.
Great. Another question we had is digital droplet PCR is a good way to validate mosaics. How is the MIPS-based approach compared to ddPCR?
So yeah, digital droplet is definitely a good way to go. However, the MIPS allow us to analyze a larger number of variants at once, and digital droplet PCR can be limited by the number of droplets per run. So we were able to validate several thousand variants in a large number of people using the MIPS-based approach. But I think if you have some smaller sample sizes or as kind of the droplet technology advances, that I think digital droplet PCR is still a good method as well.
This question is more directed towards Elaine. Is the gene expression patterns, the critical exons and brain regions, specific to mosaics or de novo mutations in general?
Yes, so the gene expression patterns that we described are specific to the mosaic mutations, and we did look at all the de novo mutations in general, but I think at a very early stage, and we didn't observe the same pattern.
And the second question for you, Elaine, is how did you deal with repeats?
Yes, so we did exclude mosaic mutations that were detected in highly repetitive regions from the Macaro paper that was describing mosaic mutations. And In addition, we did a couple of experiments as well to test for copy number polymorphic regions, and we didn't actually see any of these Mosaic mutations that fell into copy number polymorphic regions of the genome.
We had another question, which was, Rebecca, you only looked in your study was looking in the SSC, whereas Elaine, yours was looking in SSC and the ASC. Do you think that the different data sets might account for some of the discrepancy in the results that you had?
So yes, perhaps they would, but in our analyses, what I sort of glossed over was that we compared any of the observations that we detected in the SSC with the ASC as well. And so we broke them up in some of the supplementary material to show that there really isn't much of a difference between the SSC data set, or at least the analysis ran on the SSC portion of the data compared to the ASC.
I think it also just comes down to different ways of looking at the data and more of kind of our analyses. We developed our model approach, and we had different filtering strategies arriving at some different representations of our mosaic mutations present. I think particularly kind of having that high joint coverage threshold and different ways we looked at constrained variants or gene expressed variants can be maybe one rationale for the discrepancies, I think.
Another question that we had as well was, did you see any sex differences, or was that something you weren't looking for?
I think we did not explore the sex differences, but we would not expect the sex differences for the Mosaic mutations, really. I'm not sure.
Yeah, we didn't specifically explore the kind of like with the germline mutations, whether or not we saw more severe mutation, the number of more severe mutations in females versus males, but I think we observed a lower number of the loss of function mutations, so really didn't have a lot of power to kind of go into any in-depth analysis, just looking at kind of the stronger loss of functions. But we did do some analysis looking at loss of function in missense between in terms of IQ, and we didn't see much of a difference, but again, we kind of saw a low number of loss of function, so kind of underpowered there.
I wanted to ask, I'll start with you, Rebecca, and you've also, you mentioned earlier that you're starting to look towards other conditions, and I'm wondering what the next steps from these studies are for you.
Yeah, so one, we really only looked at, the single nucleotide variants, but we are also exploring insertions and deletions and kind of following up on that analysis. We are also looking into possible discordant twin families and seeing if mosaic differences in mosaic mutations between twins concordant and discordant for autism, and with some focal cortical dysplasia patients, we actually have the opportunity to look at brain tissue as patients have the dysplastic lesion resected, and we've been lucky enough to get peripheral tissue, mostly saliva, but potentially also blood, and look to see if we can detect mosaic variants in the saliva and brain if they're present, saliva and blood if they're present in the brain, and also see if mosaicism, is a contributing factor to focal cortical dysplasia.
Elaine, I have another question, which is just specifically for you. For any of your top 10 genes, did you see that they had specific functions in the amygdala?
Yes, so many of the genes that we found with recurrent, non-synonymous mosaics are indeed expressed in the amygdala, such as SMARCA4, but I've not compared if they are exclusively expressed only in the amygdala and not other regions of the brain.
Okay.
So
and Elaine, I also want to put the question to you: what you feel what are the next steps for you from this study?
So I think some of the take-home findings that we found interesting from the work is that perhaps by studying mosaic mutations, we can start to explore the genetic architecture of some of these individuals with autism, but are high functioning as opposed to the low functioning, autism spectrum, individuals affected by low functioning autism, which has been previously reported for many of the germline de novo mutations. So that to us was a pretty compelling or interesting approach to explore in the data and in other future data sets as well.
I also wanted to give you both a chance if you have questions for each other. I don't know if, Rebecca, you want to go first and ask Elaine anything about her study that caught your attention.
Oh, I just wanted to know a little bit more about the differences in the mutational spectrum. So I thought it was kind of interesting that we arrived at kind of different conclusions about the mutational spectrum. So, we found that germline mutations and mosaic mutations seem to derive from similar mutational processes, but your study suggests that they might be more driven by defective mismatch repair, and I just would be curious a little bit more, kind of how you did the stranding analysis and just kind of your thoughts on those differences.
Mm-hmm. Yeah, so we did extensive comparison between the germline mutations as well as the mosaic mutations, partly because we understand that many of the somatic mutations that were discovered in cancer genomics have different properties. For instance, like germline de novo mutations arise primarily because of spontaneous deamination, whereas many of these somatic mutations occur primarily because of differences in fidelity of the transcription-mediated repair mechanisms. So we were trying to tease apart some of these differences in mechanisms, and we did see them in the mosaic mutations that we detected from blood. For instance, there is a strong negative or anti-sense strand bias, as well as a modest but not significant excess of these mosaic mutations accumulating in regions of the DNA that have later replication timing and so forth. So then we compared the mutational properties in terms of the differences compared to the germline mutations, and we saw certain trends. The C to Gs are still the primarily dominant group of mutations that occur. However, we did see subtle changes between our group C, that is the high confidence mosaics, when comparing to all the germline de novo mutations.
And Elaine, is there anything about Rebecca's paper that you would want to know more about?
Yeah, so I think it's actually really interesting in terms to think of how the synonymous mutations could affect splicing, because that's obviously a very difficult and understudied problem. So what are some of your thoughts in pursuing some of these synonymous mosaic mutations that are enriched in the probands, and how can we start to think about, like how they affect, like some of these mutations and how they affect splicing and what could have gone wrong? And do you think that they affect splicing in a brain-dependent fashion? Like, are they specific to just brain tissue or ubiquitously in, all tissue types?
Right. So that is definitely something that we're pursuing now, is actually validating in vitro the predicted splice affecting variants. So doing like many gene assays where we see if there is inclusion or exclusion of an exon, and we definitely saw splice affecting variants really close to the splice site in some brain expressed genes. Some of the ones that we saw were ACTL6B, which is also a chromatin modifier, as well as DAM1, FBN1, and STMN1. We didn't have, a large enough, I think, sample size of some of these close splice site splice affecting synonymous variants to really get enough power to get significant enrichment in any one of these gene sets, but we saw even stronger signal, similar to what we did with the Missense, when we restricted to probands who did not have any LGDs or non-synonymous. We saw the signal increase even more so, especially in that 12.5% 50X. So I think that was kind of encouraged us even more as the probands who didn't have any other, readily or a variant that seemed likely contributing, that the synonymous variants may actually be playing a role in, some of our strongest candidates like ACTL6B are in probands who don't have any other germline mutation that would seem contributory, and are actively kind of validating that approach now. So.
Okay, great. So I think that we're coming towards the end of our time, and so I just want to thank both of you for such in-depth presentations, and also for taking the time to answer everyone's questions about both of your studies today. And so just for the audience, a complete replay of this journal club will be available on the Spectrum news site in a couple of days. The journal club is part of an ongoing series, so please check our site for upcoming journal clubs to be announced soon, and also for video archives of all of our webinars. You can also get information on upcoming webinars there. The next one is on October 18th with Connie Kasari. And I just want to thank Elaine and Rebecca again, and thank you all for tuning in.